endocytosis, the opposite form of endocytosis. So the vesicle is going outside of the cell. This is a vesicle, it wants to go outside. What is do you think that there will be some problem for the total amount of cell membrane, because in exocytosis, the total amount of the circle of the membrane of vesicle will be fused to the cell membrane. But in endocytosis, some part of the cell membrane will be detached. Is it something wrong with the total amount of the cell membrane after exocytosis or endocytosis, or no? Nothing. Why?
Because it's not changing.
It is okay. What why?
It's the same part of the cell membrane. It's not changing. It's just taking one part goes, one part comes.
Yes, because as much as is exactly right. Okay, as much as the endocytosis it will be detached, the exocytosis is they are equal. So nothing happened to the total amount of the cell membrane. Okay. And just remember that endocytosis and exocytosis, they are active transport. So we need ATP for both of them. Now we are talking about a very specific endocytosis for LDL. What is LDL? LDL is the abbreviation of low density lipoprotein. Also, we call that this is bad cholesterol. And we have also HDL. HDL is good cholesterol, it's high density lipoprotein. We have one molecule, most of it is protein, and some part is cholesterol. Okay, but in the LDL, the low density protein is small, and cholesterol is high. Because of that, LDL contains a lot of cholesterol. So it's not good because most of it is lipid and cholesterol is not. When it's inside of the vessel, it can cluster and make some atherosclerosis, okay? But in HDL, high density, most of it is protein, and the cholesterol is small. So they are, H means high, means related to protein. Low means related to protein, not to the cholesterol. Cholesterol is reverse, okay? But anyway, all the cells, for their function, they need cholesterol. Every cell can produce new cholesterol, but also they need the entry of cholesterol from the blood circulation. So, for the best function of the cell, cholesterol is necessary, but not too much cholesterol. But anyway, cholesterol is necessary, and some part of the cholesterol must enter the cell. How the cholesterol can enter the cell? This is lipoprotein. So it's LDL, lipoprotein, and it's some kind of endocytosis which is receptor mediated. Here in the blue, you see the receptor for the LDL. Only this part, after attachment to the receptor, the LDL can bring a cycle, a vesicle, and can enter and swallow by the cell. After coming into the cell, what will happen? This is the cluttering. Do you remember cluttering? But which one? Receptor mediated. This is exactly the same for entry of the iron into the cell. This is the for transferring ferritin, and exactly it's receptor mediated. Okay. But right now we will finish with carrier and different function of proteins. We come back to the slide of different function of proteins. We say that the proteins, most of them are transporter, and as the transporter, they transport channels. They are channels transporting ions. They are pumps transporting ions, but with ATP. The first one is without ATP. Okay. And carrier, they transport other things beside ions, like glucose, like amino acid. Okay. And in this way, either they are using energy, so they are active transport, the way that they are transporting is active transport. Sometimes the carriers are used facilitated transport without using any energy. Now we are talking about the receptors, another function of proteins. Sometimes the proteins, they don't want to transport anything from outside to inside of the cell, but they want to produce a signal of that substances. I will give some examples that you understand. This is the cell surface, and this is, for example, the protein or the receptor for insulin. Insulin is a hormone that it will let more glucose coming inside. When after attachment to the receptor, the glucose can enter the cell. But this is a protein kind, but this protein doesn't want to enter the insulin. The insulin doesn't want to transport inside it, but the signal of the insulin, which is letting the glucose coming inside, is important, okay? So receptors, the aim of receptors is not letting or transporting any substances from outside to inside of the cell. They want, please, if you have cell physiology, after attaching of the insulin to the receptor, some signal will be created, and after finishing of this signaling, the glucose can enter the cell. Okay? Understand? So receptors, after attaching any substances to receptors, some cell signalings will be created, and then the final signal will be created. I will explain how it can be happen. The insulin will be attached to the receptor, and then we have a carrier for glucose. Do you remember it was GLOT? GLOT is the name of the carrier. Normally, the carrier is silent inside of the cytoplasm, but after attaching of insulin to the receptor, some cell signaling will be created, and finally, at the final part, a kinase. This is the name of an enzyme will be created. This is the final part of signaling after the attaching of insulin to the receptor. What is the use of kinase for activating the GLOT and inserting into the cell surface? Do you know what is the kinase, this enzyme? They can phosphorylate other proteins, okay? So give the phosphate to other proteins. This kinase can be phosphorylated GLOT, and normally proteins after phosphorylation, they will be active. Okay, so GLOT, the carrier of glucose, which was silent inside of the cytoplasm, after phosphorylation by kinase, it will be activated, so it will insert to the cell surface and let the glucose coming inside. This is the cell signaling. So, what is the role of receptor? The role of receptor is not necessary for insulin coming inside. After attaching to the hormone substances, anything like that, some cell signaling will be created, and the final part of cell signaling will be the aim of the insulin, which is activation of GLOT here. Okay, here. Is it possible to call the insulin as ligand? Yes, it's a ligand. So hormones also can be ligand. Ligand is everything that can be attached to a receptor. Whenever, and at that time, the receptor was a sodium ion for acetylcholine. It was a chloride ion for GABA. So GABA was the ligand, and it was attached to the butt. At that time, the chloride channel or the sodium channel was the receptor. So the receptor was a channel. But after attaching to it, the gate was open and the ion. Here, there is no gate, and it's not a channel. But anyway, there is some signaling, no matter. at the final part, the aim it will appear, okay? Important cell signaling and the receptors I'm talking right now, just one, because it's the diversity of receptors is too much. But you must understand about it, otherwise when you are learning about the hormones, neurotransmitters, you cannot understand. The problem is that you cannot. So, this is the ligand, this is the receptor on the surface. After attaching the ligand to the receptor, some cell signalings will happen, and finally, what is the aim of these substances will be created. This is very simple. So here, the protein is functioning as a receptor. It's not a transporter. It's not coming inside, but it's function as the receptor. It means that some cell signaling is created for seeing what is the answer to these substances. Okay, the cell signal is the answer to that ligand, to that substances. Sometimes the receptor is located on the surface. Sometimes the receptor is located in the cytoplasm, sometimes even inside of the nucleus. For example, for the hormones of thyroid, you know, T3 and T4, when they want to activate their receptors, where is the location of their receptor? Is it outside in the cell surface, inside of the cytoplasm, or inside of the nucleus? In the nucleus, okay? For sex hormone, for example, testosterone, for estradiol, the hormone which are lipid, what is the location of the receptors? The location is inside of the cytoplasm. But for the rest of hormones, for the rest of the substances, and the majority of the receptors, they are located here in the cell surface. We can divide the cell surface receptors into very vesting part, but the most important one, we call them as G protein. G protein. So, on the top is the shape of the G protein. This is the receptor. Why we call it as G protein? This part is protein. Because in the cytoplasm, we have a complex of 3 different proteins, G alpha, G beta, and G gamma. Normally, they are attaching to each other, okay? This part is very complicated, okay? Very complicated part of the cell. But I must simplify, I will try to simplify, because you must understand this part. Otherwise, if you refer to the textbook of physiology, they are saying that this hormone is going to secrete AMP. You cannot understand what does it mean. Yes, at least a very basic and fundamental knowledge you must know about the receptors and cell signaling. This is very basic, but you must understand it, okay? But my question regarding this part in the exam is very easy, not very complicated. Okay, when there is no ligand, this is the ligand, the hormone that wants to attach the receptors. At first, there is no ligand, and these alpha, beta, and gamma of G is attaching to each other on the cytoplasmic part. And GTP, you know that GTP has one phosphate less than GTP. It's exactly like ATP, ADP. This is GTP, GDP, and GNP, okay? At first, GDP is attaching to alpha. But what will happen after attaching the ligand to the receptors? Some changing in the shape of these complex will happen. So alpha, beta, and gamma can be detached from each other. Why? Because now alpha doesn't want it will release the GDP go outside, and now it will attach the GTP to alpha. GTP is more energetic than GDP, but because it has one more phosphate, it has 2 phosphate, it has 3 phosphate. Because of one extra energetic phosphate, These energy can separate alpha from beta gamma. And the rest from here, now the alpha, which is active, starts cell signaling. Until here, there is no cell signaling, okay? But finally here, we can see the cell signaling. To see what is happening. This is the most important cell signaling for, GTP. It will create cyclic AMP and finally protein kinase A, the kinase here. Normally, most of the cell signaling is related to activation 1 kinase. Here we have the activation of kinase A. So this is the most important cell signaling for G protein receptors, which are located on the surface. I will give you one example about one hormone which uses this signaling. Okay? Do you know what is the ADH hormone? Antidiuretic hormone? Antidiuretic hormone, no? When you are drinking a lot of water, this water must go to the urine and not come inside the body. So, and there is also the opposite. Sometimes you are drinking very small water. Your body is in the lack of water, okay? So your body needs absorbing more water. How it can happen? The urination, when you are drinking very less water, your urination will be very few. And the water from the kidney will come back to the body, okay? This hormone is responsible for absorbing the water from urine. I will try to find this is in the hormone section, not here, but I will try to find the slide so you can understand what is happening. It's necessary that this is what is the tubule of the urine in the kidney. Okay? This is nephron. This is nephron, the kidney. This part, final part, is responsible. Here is urine. Do you know what is urine? Okay. Now we have lack of water. So the water here must absorb from urine to blood. And ADH, this hormone, is very important for doing it. If we see how ADH is working on this cell, I will show one magnify of these cells. This is one of these cells. This is blood. ADH is hormone, so ADH is coming here. This is urine. And we want that the water will absorb from the urine inside of this cell and then to the blood, okay? This is the role of ADH. Now we want to see that, and here we have the G protein receptors for attaching to ADH. ADH is the ligand. After attaching to receptor, this is a G protein receptor. It will activate the cell signaling will activate the protein kinase I. Now we want to see that how production of protein kinase just stop it, please.
كان لازم يدوس الثاني.
Now we want to see that what is the relationship between the production of protein kinase A and absorption of water. At first, I want to say that the water not only can directly absorb by osmosis, but we have some kind of carrier proteins for absorbing more and more water. So for water, we have two ways for absorption. One is osmosis, direct coming inside of the lipid bilayer. The other one coming through these channels. Because this channel has aquaporins. Aquaporins are very important right now because they are helping more and more absorption. Because these channels, they are protein, normally they are silent inside of the cytoplasm. For activation and coming here and helping the absorption of water, they need being phosphorylated. So, But which enzyme is necessary for phosphorylation of aquaporin? Protein kinase A. So what happened? Your body needs to absorb water because you drink a small water, small drinking, okay? Because of that, ADH is produced as a hormone coming here, attaching to G protein receptor. This is the cell signaling of G protein, producing cyclic AMP and protein kinase A. Protein kinase, they are responsible for activating the aquaporin while phosphorylation then. And after activation and phosphorylation of aquaporin, they will be activated and coming here and helping the absorption of water. So this is exactly what happened. The cell signaling of a G protein which is located on the surface of the cell. But we have another way also, the cyclic AMP and protein kinase A. This is only one cell signaling for G protein. We have another cell signaling for G protein, which is this one. This is the first one. It's very complicated. I'm talking only this part. This is the first cell signaling of G protein, cyclic AMP protein kinase A. The second signaling of G protein is producing two substances, IP3 and DAP. And the final thing is increasing the calcium. Just remember that whenever the cell uses this part, every cell, normally the smooth muscle that needs contraction, for example, the uterus. In the uterus, we have this signaling. Why? Because the uterus for contraction needs calcium. The smooth muscle, the muscles for contraction, they need calcium. So any hormone that causes the contraction of muscles, they use this cell signal and increasing calcium. For example, adrenaline, what is the reaction of adrenaline in the vessel? Decreasing the diameter and increasing the blood pressure. How? The diameter of the vessel will be less after adrenaline because of contraction of a smooth muscle in the vessel. So we have in our vessel we have a smooth muscle. Whenever adrenaline is high, your blood pressure will be high. Why? Because in the vessel we have the contraction of the smooth muscle. How it can happen? By increasing calcium, by releasing calcium. And when the adrenaline attached to the receptor, it will cause this way and increasing the calcium. The receptor for adrenaline is also a G protein receptor. So in that case, adrenaline is also a ligand. The receptor is a G protein, but cell signaling is different. It knows using this one because it doesn't need any phosphorylation of any protein. It needs release of calcium for contraction of muscle, a smooth muscle, and decreasing the blood, the vessel, and increasing the blood pressure. Okay, do you understand?
Yeah.
Just remember these two ways. Later in the hormone or any other parts in the heart, you need to understand these things. Okay. Inside of all the cells is negative charges. Okay. And outside is positive. Why we have negative and positive charges? It's not that electron, like the wire, okay? We have the imbalance between positive ions and negative ions, okay? Normally, we have less positive ion inside, and because of that, it's minus, for example, 90. And we call it as EAN. EAN means basal voltage of the cell. So if we insert an electrode inside of the cell, the other electrode outside, and then put it to the voltmeter, all cells are inside negative than outside. And this is, fixed for all cells. But sometimes, only in two cells, if you insert some stimulus, some water, for example, acid, to these cells, this is a nervous system, okay, nervous. What will happen? You see that these they are changing, okay, coming up, down. And if you put some pen to that electrode, you that the pen is showing some structure like that. This is action potential. So action potential is the answer of one muscle or one nerve to some stimulus. This action, this answer is the changing imbalance in the voltage of the cell membrane, okay? So the cell membrane in the muscle and in the nerves is not fixed. Sometimes after inserting some stimulus, it can be changed. What is this changing of EAM? We call this changing and this shape as action potential. Why the muscle or the nervous system, they are creating action potential? Because they want to send this hardcore stimulus to the brain, so the brain can understand that something is happening. So they only think that nervous system and the muscles can respond is in the form of action potential. This is action potential, and this is a kind of coding of the answer to all stimulus. But after reaching to the brain will decode it, and so you can understand that this stimulus is, for example, heat, that stimulus is pressure, the other one is acid or anything like that, okay? And the resting EAM is minus 90. Here we have the stimulus, and we see that after stimulus, little by little, we have a depolarization. Depolarization means less negative, okay? So until here, it's reached here. There must be some point that we call it as threshold, threshold. What is the threshold? Why it's very important here? Do you remember the voltage-gated ion channels for sodium?
Yeah.
Okay? Come back to that. I'm sorry that because I must all the time change it from one part to the other part. Here. Okay. They are the shape of the voltage-gated sodium channels, and we know that the gate, the upper gate, is normally closed. But when the cell is reaching to threshold, the gate can be removed. Now a lot of sodium can enter, and we can reach to this part. Here is threshold. This is minus 90. This is EAM. At rest, this is minus 60, for example. This is our threshold. Then at this part, the gate is removed and a lot of sodium can enter, and we have a sudden uplight. This is minus 35. My problem is that the ion sodium through this gate is coming after this point. What we have, here is the stimulus. From the stimulus to the threshold, we have the depolarization. Which channel is responsible? This is a sodium channel, but not this one. Because at this point, they are closed. The gate has not been yet removed. So how this depolarization, the earlier depolarization can happen after the stimulus? You understand what I'm talking?
Yes.
This is the stimulus. We have a small upgoing. This is a small upgoing and depolarization. It's necessary because it wants to reach the threshold, and after threshold, the gate will be removed, and we have a sudden huge coming inside of sodium and sudden action potential. But before that, in this part, they are closed. Why we have depolarization? How it can be depolarized? Of course, it can be sodium that can enter because the potassium is not working here. But how? Which channel? They are not. Yes.
What channel?
Sorry.
Pump.
Pump?
No.
No, pump is not pump channel. We have another kind of leaky channels. Do you remember leaky channels? The leaky channels, they don't have any gate. Okay, normally they are like. So, and a little by little, so they are leaky channels. The leaky channels, they don't have, this is the cell membrane, they are leaky channels without any bulb, without any gate, okay? But here, sometimes this diameter is very narrow, when the cell is at rest, there is no stimulus, we have narrow and very small sodium can enter the cell. But what happens after a stimulus? They will be wide, because they will be more wide like that. Why it will be more wide? Because here and here, the arrangement of phospholipid will be with double bond. So all the phospholipid with double bond will come here, okay, after a stimulus. Not they just detach the entry of the channel, and more sodium comes. So everything, this is high fluidity of the cell. So after a stimulus, some arrangement of phospholipid will be changed. The phospholipid comes close to the channel with more double bonds. The phospholipid is like that. When we have double bonds, they will be separated. Okay, so this entry will be more wide. Okay, and because of that, we have more entry of sodium. More entry of sodium means that more depolarization. But it's not very huge sodium. It's very small compared to these ones. Okay, the leaky channel compared to the gated channel, they are working very small for sodium. But anyway, they created a small depolarization. After attending to the threshold, suddenly the gate of the voltage gated will be removed, and a huge amount of sodium will come inside, and we have the sudden upward of the sodium. The sodium wants to keep continue. Right at this part, it will be finished, and it will not go further. Why? Because at this time, the other gate is closed. The bottom part will be closed, so no more sodium can enter, okay? This is from here to that part, is from there to that part. And this part, we are here. Sometimes my question is like that. On the top part of the action potential, what is the situation of sodium voltage gated channels? Okay, you must, on the top, we are here. You must say that the outgoing part is open, the inside or outside is closed, is open, the inside is closed. Okay? Here, at the threshold, we are here. And you say that most of them are open. Before threshold, at rest, we are here. Okay? Sometimes my question is like that. One example of my questions. It is responsible for repolarization at this part. Do you know?
What?
The repolarization means coming back from the positive, coming back to the minus 90. So it means that we have negative inside. But is it entry of chloride or going outside of one positive? Which one for repolarization? The chloride is coming, the negative is coming inside, or a positive is going outside? Positive is going outside. Chloride is nothing. There's no role here. But which positive? Sodium never will go outside. There is only potassium that wants to go outside. So here we have the channel for, gated channel for potassium. At this part, the potassium, the gate of potassium will open, and the potassium is going outside. Because potassium is positive, the voltage becomes back to the negative, okay? The cell is losing one positive ion. Losing one positive ion means one negative inside. They are equal, okay? Normally, when we are here, what is the shape of the at rest, okay? What is the composition of sodium and potassium at rest? You know which ion is normally high inside and which ion normally is high outside at rest? just talking about sodium and potassium. We are at rest. This is a stimulus. We are at rest before a stimulus. This is the cell.
Yeah.
Which ion is normally high outside potassium?
Sodium.
And which ion is high?
Potassium.
This is the normal shape of, ions inside and outside at rest. Okay. But during the action potential, the first phase, the depolarization phase, what is happening? Sodium is coming inside. So sodium now will be high inside in this part, and outside it's small. And during repolarization, what is happening? Potassium is going outside. Now potassium is high and inside is low. So this is the final part here. This is the final part here. At the final part, we have a cell with opposite ions. Inside, sodium is high. Outside, potassium is high. For reversing everything, which carrier or pump is responsible for coming back to the normal situation? Sodium, potassium, ATPase. So, at the final part of action potential, sodium, potassium, ATPase is working. What is the role of sodium, potassium, ATPase? It will let go 3 sodium outside
And 2 potassium come
potassium
out.
inside. So everything will be okay like the first one, okay? The potassium will high inside and the sodium will high outside, okay? So at the final part of action potential in the nervous system, all the ions, they are opposite. They are reversed. Inside, sodium is high, outside, potassium is high. At this part, sodium-potassium activation will start and everything will come back to the normal level inside of this cell. There is another part. This is here, we call it as repolarization, means coming back to the normal level of 90 minus. But here we have more negative. We call it as hyperpolarization. Which channel is enough for you?
No, I think you get over the action potential. What you actually.
No, I will start in 5 minutes. Yes. No, it's the basic of action potential. This class is for action potential.
No, I want I thought you are get over the action potential.
Okay.
Yes.
The action potential is the final part. So after that,
When we finish all of that, that's
the
it.
muscle will start next week.
What?
Muscle.
Just action potential and physiology.
Yeah. Yes, for me, action potential is the final part. Then, why more negative, more going outside of potassium. In this part, it has not been stopped. Why? Because this channel is very slow compared to sodium channel. So slowly it will go, and then come back to this. It starts to open there and it wants to come back at minus 90 like that, close, okay? But this gate is very slow. So when it reaches to the minus 90, the open is not completely closed. Little by little, potassium is going outside. And we have a more negative hyperpolarization because this channel is slow, and at minus 90 it has not been completely closed, and a little bit more potassium is going outside. More potassium is going outside means more negative inside. Hyperpolarization of this one, okay? Now we are going to start about that, to say that why here, okay, here you see the normal amount of sodium outside, sodium inside, so sodium normally is outside, potassium and is normally inside. I have one question, we are just talking about these 2 ions for action potential right now, not for any other
Material.
ions, okay. Just don't think about action potential, only think that we have some channels for sodium and potassium, and the normal amount of sodium is outside and potassium normally is inside. Just we are at first talking about sodium, not about action potential. Please pay attention because this part is very important, okay? Just thinking that sodium wants to come inside of the cell by channels, okay? We have some forces that force the sodium to come inside. Right now, I want to prove why sodium never go outside by channels. All the time wants to come inside, okay? We have some channels for the sodium, and you know that the concentration of sodium is outside. So, for every ions that wants to come inside or outside of the cells, we have two forces. One force is according to the concentration. We call it as chemical gradient. So, for transport, it's not good. For transport of any ion, we have two forces. One force is, we call it as chemical gradient. Gradient means force, okay? It's according to concentration. Which part is more concentration? And the other force, the second force is electrical. For sodium, we can show this chemical and electrical gradient by one arrow. This is arrow, okay? For sodium, at first, show the chemical gradient. Because outside is too much, the chemical gradient is from more concentrated to less concentrated. This is chemical gradient. It's according to the concentration of the sodium. Because sodium is hard outside, so sodium all the time wants to come inside, okay? But because sodium has positive charge, and we know that inside is negative and outside is positive, we have also electrical gradient. For example, for glucose, because glucose doesn't have any charge, we have only chemical gradient according to concentration. But for ions, because ions are not only they have some concentration, but also they have some charges, we have two forces. One according to the chemical, the other one according to electrical. Okay? We have another force for sodium. This is electrical. It's positive. It wants to absorb negative. So it wants to come inside. Positive absorb negative. Okay? So both of the electrical and chemical gradients for sodium is coming inside of cells. Do you understand or not? I will repeat. So it's easy. Now I prove my talk. Never sodium wants to go outside because both of the gradients is coming inside. Chemical and electrical. But now we want to show for potassium. I was telling you before that potassium wants to come inside or go outside? Outside. This is the channel for potassium. Potassium all the time wants to go outside. But now I want to prove it for you, okay? According to electrical and chemical gradient. Now just write the gradient for potassium. This is the channel for potassium. The chemical gradient is from inside to go outside. This is the chemical. It's okay. But how about the electrical? Potassium is a positive ion. Positive wants to absorb inside. So the inside wants, the other one is from outside to inside. Right, it's outside, the other force wants to come inside. What will happen to potassium finally? I'm telling you that potassium is all the time wants to go outside. It means that the chemical gradient is greater than electrical gradient. But I want to prove it by mathematics. We have some calculation mathematics that I can prove it. Okay. Previously, I was talking about the the EM. EM was basal voltage of membrane, okay? And it was, for example, minus 90 for nervous system. But we have another voltage. It is related for each ion. This is the equilibrium voltage of that ion, and we call it as Nernst voltage for different ions. We have Nernst voltage for sodium, we have Nernst voltage for potassium. What is it? What does it mean? It means that if the voltage of the membrane reaches to that voltage for sodium, sodium is in the equilibrium, no movement of sodium. So every sodium or potassium, they want to move in accordance to their Nernst equation. We have for one calculation for reaching the Nernst equation. This is Nernst equation for each ion. The calculation is that the log of that ion concentration inside, for example, sodium inside, then the sodium outside. So at first we have the concentration, we must have the concentration of sodium inside, then divided to sodium outside concentration, then we log it, and then we multiply to that one, okay?
Yeah.
We don't use, this is just formula. You must understand that the next equation is that. We never ask you to calculate the logarithm like that. We have the next equation for different ions, and I will give you for like that. Sorry. This is the actual, the first Nernst equation. It's very difficult. So it's not here, I will give you. The Nernst equation for sodium or potassium, I will write it according to this formula. So we have the formula. According to this formula, I can calculate the Nernst equation for sodium and potassium. The Nernst equation for potassium is, for example, minus 1. Okay. The Nernst equation for sodium is minus 65. What does this mean? It means that in potassium wants to move in order that EM, which is normally, for example, minus 90, reach to that. After reaching to that number, which is the Nernst equation of potassium, potassium is okay. It doesn't want to move anymore. So it's equilibrium for potassium. Sodium, every ion has a Nernst equation for itself. Sodium for sodium is this number. It means that after reaching minus 90 to that one, sodium is okay, and no more movement of sodium, okay? So, for potassium, whenever your ion, the chemical gradient is opposite to electrical gradient, exactly like potassium, and you don't want what finally happened to potassium, you must to compare the Nernst equation of potassium with EM. And then after that you understand the direction of movement for potassium. This is the Nernst equation for potassium is more negative. And this is an ideal number for potassium. It's not reality. The reality is minus 90, which you can be recording, okay? So the reality, this is the reality. The actual number of EM. The Nernst equation of potassium, this is ideal. So which one normally wants to reach to other one? This is reality, this is your life, this is your ideal life. You want to try to reach what you are really to the ideal. Okay, so the potassium wants to move in order that the reality reach to the ideal form. Okay? We know that potassium is positive. So how potassium as a positive ion wants to make the number more negative. This is the reality number of the EAM, of the voltage of membrane. This is the ideal voltage. This is more negative. Potassium is a positive ion. When it wants to make the voltage of membrane more negative, it must go outside. Okay? So you understand that which gradient is greater, the chemical gradient, that one which is forcing potassium going outside. So come back again. You want to understand that one ion wants to transport either inside or going outside of the cell. At first, exactly, the first thing that you will do is to drag the chemical and electrical gradient. If both of them show the one direction, like sodium, everything is okay. You say that this ion is, for example, coming inside, like sodium. But if the direction they are opposite, one showing inside of the cell, the other one is showing outside of the cell, you cannot decide which one is greater, like potassium, okay? In that case, you must compare the Nernst equation of that ion with the EAM. The EAM is the reality and the Nernst equation is ideal. The ion wants to reach the reality to the ideal. In that case, you understand after comparing which gradient is greater. I will give you 1, 2, 3 examples for understanding better, okay? And also in the exam, this is also one of the questions in the exam. In the exam, I will tell you that there is one ion, for example, X. This is the ion X. The charge is negative. The concentration outside is 10 times, inside is smaller, okay? I will ask you that this ion is coming inside or going outside. The first thing is to drag the arrow, chemical and gradient. The chemical is according to concentration. Outside is much more concentrated, inside is less. So what is the gradient from
Get
more
into
concentration?
from the high concentration.
This is the chemical gradient. Hi, what electrical? Inside is negative, outside is positive. So negative absorb positive, so it will go outside. This is electrical. So in that case, you cannot tell the direction of movement because they are opposite. Here, when they are opposite, you need to compare the EAM and Nernst equation for that ion. I will give you the number of these two, okay? At the part, I will say that if you understand that EAM for this is minus 90, and Nernst equation for that ion is, for example, minus 103 millivolts. So just tell me which direction is greater, that one or that one. The Nernst equation is ideal. It's not real. The EAM is real. The ideal, which one wants to reach to the other one? Real wants to reach the ideal. Okay? So the ion wants to transport in order to make the cell more negative. The ion is negative. How it can make the cell membrane more negative when it's negative? Come inside. So this one is greater. So the ion is coming inside. Okay? Just if this EAM, this equation was, for example, positive, for example, like that. This is ideal, this is reality. The ion is negative. It wants to make the EAM positive. How it can be happen that a negative ion makes positive, it must go outside. So this one is greater. Do you understand? So at first, I will give you one example in the exam. I will draw something like that. And the first thing that you must know, just draw these two chemical and electrical gradients. If they show the same, you are okay. But sometimes you cannot understand. Sometimes your arrow is it's better to compare this because after comparing, they must be according to these different arrows, okay? But if they are opposite, you need these two. I will just give you the example for sodium. For example, the sodium is outside is higher, inside is smaller. So for sodium, if the chemical gradient is coming inside, and because it's positive and inside is negative, the electrical also show coming inside. So both direction is showing inside. But because I will give you also these numbers. Even if you are not sure about these directions, it's better to compare these two. Sometimes you are drawing the arrow, but you are having some mistakes, okay? It's better, both of them must be in accordance to each other, okay? After showing the drawing of these two right, then compare these two together. This is the reality, this is the ideal of the sodium. It wants to reach to a positive. How it's possible for sodium as a positive ion to make the EAM, the part of the charge membrane to positive by coming inside. So it's okay. Both of them they are not opposites. Everything they are confirming the other thing. Okay. I think that I need some half an hour while finishing your class. Maybe I will arrange, okay, just a very small part and for reviewing, okay? I think that it's better maybe during the next week or maybe after that.
Anytime you want.
The time after the Dr. Azizi, maybe half an hour after Dr. Azizi, just please study everything that I was mentioned. So I will bring also some of the example of my questions and I will finish this part. Because I want to be sure that after reviewing, you understand everything. So I need just half an hour, maybe less than that, but we review everything that I was telling you, and I will give you some of the previous example of my previous classes, so you understand that you are okay for your exam.
Okay.
Do you know what is the date of your exam?
Yeah. Yeah, maybe.